So today's Insta lecture is on classification of hemolytic anemia, and the best approach to approach hemolytic anemia is to divide it into two groups. One group, I would say, intrinsic causes which are leading to hemolytic anemia. Another is extrinsic causes. Intrinsic causes, I mean the intracorpuscular causes, the problems which are inside the RBC. Now, if you look at RBC is the simplest biological cell possible, and basically it's a bag, donut shaped bag, which has made of three components. One is the covering of that bag, the RBC membrane. Second, there are a few enzymes are there for their survival. And third is that they have a lot of hemoglobins inside that. So basically, it's a bag which is full of hemoglobin. Now, if you want to look at the intrinsic causes of hemolytic anemia, basically you can divide them into three groups of causes actually. The first group is due to some defect in the RBC membrane that is leading to hemolytic anemia. And under this group of diseases would come hereditary spherocytosis, hereditary elliptocytosis. Mostly they are hereditary, as their name suggests. And sometimes there could be acquired defect also of the RBC membrane. Classic one is PNH, paroxysmal nocturnal hemoglobinuria. I would like to discuss them in the coming Insta lectures. Second group of diseases would be due to some enzymes which are there in the RBCs if they are deficient. Classic one is G6PD deficiency, glucose 6-phosphate dehydrogenase deficiency, which leads to acute hemolytic anemias when they are exposed to drugs or infection. And the third group is usually due to some problem in the hemoglobins. We usually call them hemoglobinopathies also. And sometimes the hemoglobin, they are not produced in adequate amount. They are quantitative hemoglobinopathies like thalassemia. And sometimes there could be qualitative hemoglobinopathies like sickle cell anemia. That means the sickle cell hemoglobins are quantitatively normal in amount. The total amount of hemoglobin is normal, but they are qualitatively is not like the normal hemoglobin. They are kind of defective hemoglobin. So in summary, in a nutshell, there are three groups of intrinsic hemolytic anemias. One is due to some defect in the membranes, one could be due to some defect in the enzyme, and one could be due to defect in the hemoglobin. And we could name them membranopathies, enzymopathies, and hemoglobinopathies. These all are intrinsic or the intracorpuscular causes of hemolytic anemia. Now, if you approach the extracorpuscular causes, that means there is no problem inside the RBC, but the problem is coming from the outside, and that is leading to hemolytic anemia. And by the way, hemolytic anemia means that in those anemia where the RBC's lifespan is very much shortened, that normally they're supposed to live for 120 days, but they usually live in hemolytic anemias on average less than 120 days. That's the basic idea of hemolytic anemia. So it's like we are supposed to live for, on average, currently 70 to 80 years. But if we have a premature demise at the age of 20, 25, that's I think a kind of analogy to explain it. Now I'm going back to the extrinsic causes of the hemolytic anemia. Extrinsic cause of hemolytic anemia could be due to something extrinsic. Like it could be due to autoantibodies which are produced outside, and they're coming to RBC and causing destruction of the RBC. Like SLE, there is a auto antibodies which are produced, they're causing destruction of RBCs by type 2 hypersensitivity reaction. That's a classic example of extrinsic causes leading to hemolytic anemia. Second important cause would be, there are many causes actually. It could be due to infections like malaria, it could be due to trauma. Important one, I would say, MAHA, that is microangiopathic hemolytic anemia. And microangiopathic hemolytic anemia, as the name suggests, it's made with micro plus angiopathic plus hemolytic anemia. So typically angio means blood vessel. So typically there is a pathology in the micro means small, angio means blood vessels. What is that pathology? The pathology is this, that there is usually some kind of thrombi deposition is seen, thrombus formation is seen there. And this thrombus is within the lumen of the small blood vessels. So you can think in this way that RBCs are coming at a very high speed in a blood vessel, and they get hit on a bump in an obstruction inside the lumen of the blood vessel, inside the caliber of the blood vessel, and they rupture. It's just like the example I gave actually, that it's like a high speed car, like a Formula One car that's coming and hitting the thrombi, and as a result there's an accident and the RBCs get deformed, and that results in hemolysis. And as this hemolysis is occurring in small blood vessels, we call this microangiopathic hemolytic anemia, and those ruptured RBCs are called cystocytes. So all these things I am going to discuss in the coming lectures also. So basically what we understood, that there are two groups of causes of hemolytic anemia. One are intracorpuscular or intrinsic causes, another are extrinsic causes. Intrinsic causes could be summarized into basically three groups. One is due to some membrane problem. The examples are hereditary spherocytosis, hereditary elliptocytosis, and paroxysmal nocturnal hemoglobinuria. It could be due to some enzyme defect, enzymopathies, like classic one is G6PD deficiency or pyruvate kinase deficiency. And third would be due to some problem in the hemoglobins, like sickle cell anemia, hemoglobinopathies. And other major group is extrinsic or extracorpuscular causes, where the problem is coming from the outside, like infection, like trauma, like microangiopathic hemolytic anemia, like autoimmune hemolytic anemia. So this is the classification of hemolytic anemia in a nutshell.